Unit 1, Intro to AP Biology. The fundamentals of biology rely on the ability to interpret information from labs. When setting up labs, it is important to know what the following terms are: independent variable, dependent variable, null hypothesis, and alternate hypothesis. Let's look at these terms in the context of a lab. One experiment could be to see how the salinity of water affected brine shrimp hatching. In this experiment, the salt would be the independent variable and the hatch rate would be the dependent variable. The independent variable impacts the dependent variable. In this same situation, the null hypothesis would be that salinity has no impact on hatch rate, and the alternate hypothesis would be that salinity can affect the hatch rate. When conducting labs, it is also important to consider the scientific method. The scientific method has 7 steps, which are to observe, research, form a hypothesis, test the hypothesis, analyze results and draw conclusions, report your findings, and conduct more research. The scientific method is important in biology because it helps scientists gather evidence and validate results to their experiments. Levels of organization in biology help us see and understand how organisms function within a biosphere and what they are composed of. They are arranged from most to least complex, and there are 10 levels. There's a biosphere, ecosystem, community, population, organism, organ system, organ, tissue, cell, and molecule. As you can see, these different levels sort of fit in a pyramid going from most to least complex. Evolution is the process of change that has transformed life on Earth. Evolution is gradual and is to better adapt species to their environment. Evolution is shaped by a number of mechanisms, including selective pressure, sources of variation, and random effects, such as genetic drift. Prokaryotes and eukaryotes are 2 different types of cells. The biggest difference between prokaryotes and eukaryotes is that eukaryotes have a nucleus, while prokaryotes do not. Bacteria and archaea are prokaryotes, while eukaryotes are protists, fungi, plants, and animals. Here are some additional differences between the 2 types of cells. One important difference is that eukaryotes have linear DNA, while prokaryotes have circular DNA. This is important as it affects the replication process. For something to be considered living, it must exhibit the following 7 characteristics: growth and development, energy metabolism, homeostasis, adaptation, response to stimuli, be composed of cells, and reproduce. There are many hypotheses about the origins of life. One possible hypothesis for the origin of life is that life arose gradually from inorganic molecules with building blocks like amino acids forming the first and then combining to make complex polymers. In the Miller-Urey experiment, the first evidence was provided that organic molecules needed for life could be formed from inorganic components. Stromatolites, which are, in many cases, formed by the growth of layer upon layer of single-celled microbes such as cyanobacteria, are the first fossils and can help us identify the origin of life. Charles Darwin is one of the most well-known contributors to the ideas of evolution and natural selection. Darwinism is a theory of evolution with which Darwin used the term or phrase descent with modification to describe natural selection. Natural selection is the process in which better adapted individuals are more likely to survive and therefore reproduce. There are 3 conditions that describe natural selection. Individuals with certain heritable traits have higher survival and reproduction than other individuals. When environmental changes occur, new species may arise, and the process increases the match between organisms and their environment. Studying phylogeny helps us see how animals are related. Looking at branch points and where the different branches go helps us see common ancestors between organisms.